The topic that I have chosen, knowing that it is an heterogeneous group, so that my presentation should benefit all of them. So in that context, though it is a botany topic, but I made it in such a way that it will benefit every participants of this refresher course. And I hope everyone will take some message from this today's presentation on fungal biology and biotechnology. Thank you very much, madam. I welcome all the participants for this online presentation of my talk on fungal biology and biotechnology. Hope
Good
everyone is
afternoon, sir.
hearing.
Good afternoon, sir.
Good afternoon. Good afternoon.
Good afternoon, sir.
Good afternoon, madam.
Good afternoon, sir.
Good afternoon, sir.
Good afternoon. I welcome all of you. Hope you are enjoying the refresher course, online mode of refresher course. We are not happy with this mode, but somehow we have adjusted to this online mode. But you will have a comfort of hearing special lectures, sitting at house, sitting in your college. This is one advantage. So hope you will make use of this opportunity and enhance your knowledge so that you can use this knowledge for enriching your student at the college and the university level. So I welcome once again for the presentation. So as you all know, the living world we are talking about, we had only two kingdoms, kingdom Plantae and kingdom Animalia. So much before the discovery of microscopes or microscopic organism, the naturalist, then biologist, divided the living world into two major kingdoms, the kingdom Plantae and kingdom Animalia. This classification was based on mode of nutrition, plants with autotrophic mode of nutrition and animals with heterotrophic mode of nutrition. So we had two kingdoms. Then in the 15th century onwards, 16th century, microscopes were discovered, microscopic organisms were also discovered, and therefore it was very difficult for biologists to group all the newly discovered microscopic organisms either with the plant kingdom or with the animal kingdom, because the organisms that were discovered later in the 16th century, they were distinctly different from plants and animals, and therefore it was the turn of E.H. Haeckel in 1869. He introduced the third kingdom known as kingdom Protista to group all these microscopic organisms, and his classification is now known as three kingdom classification. You are all familiar with this system of classification, the three kingdom classification. It was a E.H. Haeckel, a student of Charles Darwin in 1869, he proposed. When this classification was proposed, it was, though it was appreciated, it has never been universally accepted, because among the group of microscopic organism that were included in the third kingdom Protista, they were all different from each other. They never shared common properties with among themselves or with the plant system or with the animal system. So therefore, it took nearly another 100 years. It was the turn of American biologist Robert H. Whittaker in 1969 divided the Protista into two kingdoms again, the kingdom Monera, kingdom Protista, and he also erected another kingdom called third kingdom called fungi. Okay, and he placed the fungi in between plants and animals. He treated the third kingdom fungi along with the plants and animals. So the credit goes to Robert H. Whittaker in 1969 for having proposed this five kingdom classification. This five kingdom classification was based on, one is cellular organization at the lower level. That means kingdom Monera and kingdom Protista were classified based on cellular organization, whether the organisms are unicellular or the prokaryotic or unicellular eukaryotic. So Monera therefore included the prokaryotic organism, unicellular prokaryotic organisms, and the protista included unicellular eukaryotic organisms. He erected the third kingdom, fungi, in between Plantae and Animalia, treating along with plants and animals because fungi were also multicellular in nature. Another important characteristic feature, criterion, the Robert H. Whitaker used to differentiate these three kingdoms was the mode of nutrition. Fungi with absorptive mode of nutrition, and plants autotrophic mode of nutrition, and Animalia with ingestion mode of nutrition. The classification system, five kingdom classification system proposed by Robert H. Whitaker was appreciated and universally accepted. Only recently, in 1990, the another system, what we call three-domain system, was classified. According to this, the proponents of this classification, see, Carl C.R. Woese, in 1990, he divided the all living microorganism, all organism into three major domains: domain, bacterial domain, archibacterial domain, and eukaryotic domain. So this classification was mainly based on 16S ribosomal RNA content analysis, and he wanted to establish a phylogenetic relationship among the living organism, and therefore this is the present day, the well accepted classification system of the living world. Look at the eukarya, the eukaryotic organism. At the tip of this, we have a third kingdom called fungi, and these fungi have been placed between animals and plants. So even Carl C.R. Woese treated fungi with highly evolved animals and plant system that shows that fungi are also very important and important group of organisms on par with plants and animals. So, dear friends, today I am going to talk about why these fungal third kingdom fungi are very important in our academics and also research, and how these fungi have shaped the world research in molecular biology and biotechnology aspects. So I will cover some of the very interesting aspects of fungi in the next 1.5 hours. So let us try to understand what are these fungi. ಸೊ ಕನ್ನಡದಲ್ಲಿ ಶಿಲೀಂಧ್ರಗಳು ಅಂತ ಕರಿತೀವಿ, ಶಿಲೀಂಧ್ರಗಳು ಅಂತ. ಸೊ ದೀಸ್ ಫಂಜೈ, ದ ಡೆಫಿನೇಷನ್ to define fungi was very difficult for some time, and later it was the turn of Professor C.J. Alexopoulos, a well-known mycologist from Michigan State University. He was a professor of mycology, professor of botany, and he was teaching mycology. He exclusively worked on fungi, especially slime moulds. He also wrote a book on introductory mycology. This was one of the book in 1960s and well-read book, and this book was written by C.J. Alexopoulos, a professor of mycology at the Michigan State University. He gave a definition, and that definition even now holds good in most of most of the teachers in universities and colleges. They use this definition to define fungi. So fungi are eukaryotic organisms, that means highly evolved organisms, spore bearing. They produce the spores, a small microscopic structures called spores through which they reproduce. In addition to that, they are heterotrophic. They does not contain chlorophyll pigment. Therefore, because they are achlorophyllous in nature, and they absorb all the essential nutrient, water and essential nutrient, through absorptive mode of nutrition. That means the body of the fungus is made up of filamentous thread-like structures. We call them as hyphae. Through hyphae, they absorb all the essential nutrient from the surroundings, from the ecosystem. So, and these hyphae, filamentous thread-like structure, they are made up of cell wall, and it is in turn made up of chitin. So, this is how the fungal group are defined academically. To go further into this, these are the eukaryotic filamentous branched spore-bearing organisms. Once we thought that they are all spoilage organisms. They are responsible for causing spoilage of food items and also responsible for causing diseases in humans and animals and also plant. But it is not true. Okay, so of course they lack chlorophyll pigment, they are heterotrophic in nature, they absorb the essential nutrient through the surrounding, through their general body surface. So very interesting feature of fungal cell wall. They different from plants in having cell wall made up of chitin. Only a few are fungi which are having both chitin and cellulose, but majority of them are cell wall is made up of chitin. So that's why they are distinguished from plant cells. They reproduce by small spore-like structures. They reproduce by two different methods: asexual method as well as sexual method. By these spores, we can also classify them into different groups. I will come to that a little later. They are very diverse group of organism, very unique group of organism, very ubiquitous group of organism. They are distributed in every ecological niche. And today there are about 80 to 1,20,000 species have been described. Okay, out of 3.8 million fungi, they are being estimated from different methods. But today we have identified and described only 80,000 to 1,20,000 species. So the mycologists, they predicted that these fungi-like organisms originated around 600 million years ago. Around 400 million years ago, they migrated to land and they started living as on a terrestrial mode of life. The study of these kind of organism, the subject we call it as mycology. So what is the important role the fungi are playing in the ecosystem? The most important role is the decomposition of the organic matter. Every day, every human beings are also, and also plants, when they die, there is a lot of lignocellulosic waste gets accumulated. We, the human beings, also generating so much of waste. If the fungi do not decompose this organic matter, it will be a huge pollution problem. Okay, so fungi are the very important microscopic invisible creature. They carry out the degradation process very effectively, and therefore they are very important partner of our ecosystem. Therefore, they decompose the organic matter, and thereby they give back all the essential nutrients back to the ecosystem, so that all other organisms make use of these nutrients, okay, very effectively. And this is the role, the 95, 99%, 97% of the fungi, they do this decomposition work, and in that way, they are very useful in our ecosystem. The second important thing that fungi, they exist in three different, they are present, and they play a very important role as symbiotic organism, mutualistic organism, and saprophytic, and some of them are pathogenic organisms. As I told you, 97% of the fungi are saprobic in nature. They play a very important role in decomposition of the dead and organic matter. The remaining fungi, some of them live with other organisms. Some of the organisms live with plant system, some of them live with the animal system, and they lead a symbiotic relationship. They are called symbiotic fungi. Okay, we will discuss some of them later. Some of them, both the partners will be benefited. Such fungi are called mutualistic fungi, but there are other fungi which lead a parasitic mode of life, where they parasitize not only plant, but also humans and animals, even rotifers, aquatic animals. They are all infected by some of these fungi, and they cause diseases also. So in that context, so fungi are also very important, but the parasitic nature of fungi is restricted to 1%, maximum 1%. The other important role fungi are playing in our day-to-day life, because fungi are the important source of food. We don't believe that fungi provide food for us. But today we are using fungal cells as alternative food source for our human and animal food. So that we will discuss it little later. So they are the wonderful source of food, wonderful source of fuel, wonderful source of fertilizer, biopesticide, and several thousands of metabolites. And these metabolites have been, now we are using as pharmaceuticals in our drug gums. In addition to that, with the emergence of molecular biology and biotechnology in 1980, now we are using fungal cells as factory cells to produce the compounds of high economic value. Therefore, biotechnology is going to play a very important role. In addition to that, fungi are also very important in bioremediation, particularly in cleaning the polluted environment in our ecosystem. Fungi are the important organism, and they occupied an important place in both the classical and applied genetic research. They have been used as model organisms, and many of them have been used as factory cells to produce the products of high economic value. And some of the fungal organisms are also used to know the pathogenicity. How do they cause diseases, humans and plants? And very recently, the eukaryotic cell, fungal cells, such as yeast, can be used to produce vaccines. Some of the viral genes can be expressed, some code protein genes can be expressed in Saccharomyces cerevisiae, and therefore we can, these Saccharomyces cerevisiae yeast can also be used to produce vaccines. So in that way, the fungi are playing an important role in classical and applied genetic research. Why they are very important in genetic research? Because there are some features which are unmatched in other organisms, unmatched in other eukaryotic organisms. Fungi are very easy to grow. They can be cultivated in the laboratory. Majority of them, we can cultivate them in the laboratory on easily preparable media, and they have a short life cycle. Okay, short life cycle and amenable to biochemical studies. If one chemical is not available, they make use of the other chemical as source of energy, and they are able to grow. And the majority of the life cycle they have applied in nature, and therefore they are easy to mutate and see where the mutation is going. Okay, so that can be detected. They have a both asexual and sexual stages, so that segregation, recombination, and other parameters can be assessed and studied. We have very important model organism like Saccharomyces cerevisiae, that is the budding yeast that we use in our bakeries to prepare all the bakery items. In addition to that, it is pharmaceutically very important. We will discuss it a little later. Another, the equally important model organism is Saccharomyces pombe. It is called the budding yeast and fission yeast. Saccharomyces cerevisiae is called budding yeast. Saccharomyces cerevisiae is called the fission yeast. The another important organism, Neurospora crassa, Beadle Tatum, American genetist, they proposed a one gene, one enzyme hypothesis. A single gene is responsible for synthesis of one enzyme. That one gene, one enzyme hypothesis was conducted in an organism called Neurospora crassa. It is an ascomycetes fungus, and it is also it is called Cinderella fungal world, and that's why Neurospora crassa is an important in fungal genetics. Another organism that has been widely used is Aspergillus nidulans. Aspergillus nidulans, for various reasons, this is another organism. It is used in genetic research. So there are many people who have contributed immensely for understanding the science of fungi, other called mycology. As I told you, study of fungi is called mycology, and it was P.A. Micheli. P.A. Micheli, he was a noted Italian botanist and professor of botany at the Pisa University. He is called father of mycology. What he has done, he was the first person. He showed that the spores are nothing but seeds of fungi. And he was the one who cultivated these spores. He sprinkled these spores on a slice of watermelon, and he obtained the fungal colonies on a natural medium. Today, we have many different media are available, culture media are available. We can cultivate some of these fungi in the laboratory. We can produce several metabolites, but the credit goes to P.A. Micheli. He was an Italian botanist for having isolated the fungus and cultivated the fungus on a slice of watermelon. And that is why he is aptly be called as the father of mycology. He also wrote a book called Nova Plantarum Genera, which documented the detailed description of many fungi and how he has cultivated all of them. So in that context, the credit goes to P.A. Micheli. He is called father of mycology. In India, we know that India was ruled by Britishers. Okay, so therefore, the science of mycology was not developed. And only after 1947, only after independence, systematic study of fungi began. Much before that, when we were in the British era, the government of Royal British Kingdom, Kingdom, they appointed a British mycologist called E.J. Butler. He visited India in 1905. He was in India for nearly 20 years and he wrote a book called The Fungi of India. This is one of the first book that is available on the fungal world, the fungi that are documented from Indian soil. And he has also wrote a book on fungi and diseases of plants, and he is called father of Indian mycology, and he established an Imperial Agriculture Research Institute at Pusa and also laboratory to culture these microscopic organisms called fungi, and he is truly called father of Indian mycology. After when he has left India, there are many other mycologists who contributed immensely for understanding the science of mycology in India. Coming to this third kingdom, fungi. So we have five kingdoms according to Robert H. Whitaker: Kingdom Monera, Kingdom Protista. In between plant and animal kingdom, we have third kingdom, Mycota, otherwise known as fungi, otherwise known as Mycota. Same, say one and the same. Fungi or Mycota, one and the same. The third kingdom, Mycota, is basically divided into two divisions. One is Myxomycota. The second division is called Eumycota. So just for those who are interested in understanding the fungal world, with this one particular slide, I try to explain why they are very important group, how they have been classified. So coming to the division one, Myxomycota. Myxo means false. Mycota means fungi. They are false fungi. I told you the definition of fungi. The definition of fungi stated that fungi are filamentous organism or thread-like organisms. Okay, filamentous are thread-like. But in Myxomycota, these organisms are not filamentous, but that's why they are called false fungi. They looks like a plasmodia or pseudoplasmodia like structures. Plasmodia or pseudoplasmodia like structure. So that's why they were referred to as Myxomycota or false fungi. So their importance, we will discuss it little later. Another division of mycota is called Eumycota. Eu means true. Mycota means fungi. They are called true fungi. That means they are filamentous, thread-like structure. Body of the fungus is made up of filamentous, thread-like structures. Filamentous fungi called Eumycota. So this division Eumycota is divided into two types of fungi, lower fungi and higher fungi. So lower fungi, they are aquatic in nature. They are aquatic mode of life. They survive in water bodies. They produce a spores called zoospore, motile spores. So under that, we have three categories of fungi: chytridiomycetes, oomycetes, zygomycetes. We have another higher groups of fungi. During the course of evolution, fungi migrated from aquatic mode of life to terrestrial mode of life. They colonized the land. So under that, we have three groups of fungi called Ascomycetes. They are called sac fungi, Basidiomycetes, club fungi. The another group, artificial group called deuteromycete, otherwise called mitosporic fungi. So this is the entire fungal kingdom. This is how mycologists classified for understanding the fungi. So one of the important mycologists called Jeffrey C.J. Ainsworth, G.C. Ainsworth, he has classified the kingdom, third kingdom fungi into Myxomycota and Eumycota. This I have already told you. This is, kingdom fungi. Mycota is divided into two divisions, Myxomycota and Eumycota. Myxomycota means false fungi. Eumycota means true fungi. So under Myxomycota, they are called slime mold. They are not filamentous thread-like bodies, but they are plasmodia or amoeba-like structures. But their life cycle resembles fungi. That's why they have been treated along with the fungi. We have four classes: Acrasiomycete, Hydromyxomycete, Myxomycete, Plasmodiophoromycete. As I mentioned, under Eumycota, Ainsworth divided 5 divisions: Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and divisions are very important in each group. Coming to the first one, Myxomycota. As I mentioned, Myxomycota means false fungi. The body of the fungus is not filamentous thread-like. On the other hand, the body of these Myxomycota, members of the Myxomycota, is plasmodial in nature or pseudoplasmodial in nature. Okay, so that's why they have not been treated with other true fungi, Eumycota, but they are very important component of fungal world. Look at this. As I mentioned, they looks like a slime mold, slimy in nature. Some of them are the Fuligoseptica. Look at this. It looks, some of them, these are the, very important component of forest ecosystem. If you visit forest, some of these slimy nature of, fungi, you can notice in the wood or decaying wood, and these fungi can be studied. Look at this. This is another fungus called Physarum. So you are all biology student. You know the importance of mitochondria. The mitochondria was first isolated from this organism, Physarum falciparum. If you look at the cell biology book, they will tell you the history. This is a very important fungus from which mitochondria, eukaryotic mitochondria were isolated, and mitochondria were studied, and protoplasmic streaming. All these things have been studied by using this fungus, Physarum falciparum. This is one of the model organism that we use in cell biology. This is another slime mold called Dictyostelium discoidum. Okay, this is a single-celled organism. Okay, and it produces a small cells called social amoebae, and all these social amoebae, they come together and develops into a single-celled sporophore. Okay, at the time of reproduction, and this organism is used in morphogenetic studies. So this is also a wonderful model organism used to study the the developmental biology aspects. Coming to Eumycota, so Eumycota includes true fungi. Why they are true fungi? Because body of the fungus looks like filamentous thread-like structures. They are called hyphae, and these hyphae are made up of cell wall, which is in turn made up of chitin. And these hyphae reproduce asexually and sexually. Under this Eumycota, true fungi, we have 5 divisions have been made. One is called Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina, and Deuteromycotina. Coming to the first one, Mastigomycotina, please try to understand. Mastigo means flagella, flagellated cell. Mycotina means fungi, flagellated cell. Members of this group produce flagellated cells, flagellated zoospores, because majority of them are aquatic in nature. When they produce spore, these spores must move from one place to another place. So these spores have been provided with flagella, either whiplash flagella or tin cell type of flagella, and therefore these fungi are identified based on motile cells. All these motile cell producing fungi have been included under chytridiomycete, hypochytridiomycete, and oomycetes. So the Masticomycotina includes the flagellated zoospore producing fungi, zoosporic fungi or aquatic fungi. So which include chytridiomycete, hypochytridiomycete and oomycete. Some of them are pathogenic. They cause diseases. So that's why they are very, very important. So these are, look at the members. So one is Chytridium, Allomyces, Phytophthora. This is a late blight disease. Tada ಅಂಗಮಾರಿ ರೋಗ ಅಂತೀವಿ ಪೊಟ್ಯಾಟೋದಲ್ಲಿ ಕೊಳೆಯುವಂತ ರೋಗ ಪೊಟ್ಯಾಟೋದಲ್ಲಿ ಒಂದು ಸೀರಿಯಸ್ ರೋಗ ಬರುತ್ತೆ ಲೇಟ್ ಬ್ಲೈಟ್ ಡಿಸೀಸ್ ಅಂತ ಕರಿತೀವಿ ಇಟ್ ಈಸ್ ಕಾಸ್ಡ್ ಬೈ ಫೈಟಾಫ್ಥರಾ ದಿಸ್ ಫೈಟಾಫ್ಥರಾ ಕಮ್ಸ್ ಅಂಡರ್ oomacytes ಅಥವಾ ಮ್ಯಾಸ್ಟಿಗೊಮೈಕೋಟಿನಾ ಅಂತ ಕರಿತೀವಿ another group of fungi called zygomycotina. These fungi produce zygospore, sexual spores known as zygospore. So you can see the zygospore here. Okay, majority of the fungi produce zygospore. So that is why the name zygomycotina. So under this we have two classes are there, zygomycetes and trichomycetes. Zygomycetes are the fungi like bread mold, your rhizopus or mucor. All these fungi are very important and they easily grow on substrate containing little moisture content. Many of them are biotechnologically important. Many are used for production of acids, so enzymes. So that's why biotechnologically very, very important group of fungi. As I mentioned, there are model organisms are also there. One such model organism is called Pylobolus. It is otherwise known as hat thrower. Look at this fungus. At the tip of this sporangiophore, there is a black cap-like thing. It looks like it is a sporangiospore, black sporangiospore. This black sporangiospore is, dispersed to a greater distance when the sunlight falls on this photosensitive sporangiophore. So that's why it is called hat thrower fungi. So this fungus is used in the laboratory just to understand spore liberation in fungi, how fungi liberate their spores to a greater distances. It is an important model organism. This wonderful fungus, it can be used to study the spore liberation in fungi. The third division of Eumycota is the Ascomycotina. So otherwise known as sac fungi. Sac means these fungi produce sexual spores known as ascospores. These ascospores are enclosed in a specialized sac-like structure called ascus. That is the name asco. So look at these various types of fruiting bodies. Inside this fruiting body, these sac-like structures will be there. Inside the sac-like structure, 4 to 8 ascospores will be there. That is why they are called Ascomycotina. So any fungi which produce these ascospore producing structure comes under Ascomycotina. So this is how mycologists classify fungi under Ascomycotina. So we have Saccharomyces cerevisiae. Baker yeast comes under Ascomycotina. It is a wonderful organism used in bakery industry, wine production, beer production, and alcohol production industries. Another organism comes under Ascomycotina is the Neurospora. The one gene, one enzyme hypothesis and genetic recombination and tetrad analysis has been studied in this organism. We have a highly prized one of the mushroom called Marcella. Very costly mushroom, okay, and it also belongs to Ascomycotina. The biotechnological aspects of some of these fungi, we will discuss it little later. Coming to another group of fungi under Eumycotina is the Basidiomycotina. These fungi, they produce spores called basidiospores. Sexual spores known as basidiospore. Basidiospore producing fungi. They always grow in group. That is why these fungi are called club fungi. They always in 20 number, 30 or 100. They grow in groups. That is why they are called club fungi. So valuably very important fungi are grouped under this. We have edible mushroom, we have medicinal mushroom, we have poisonous mushroom, and in addition to that, we have disease-causing basidiomycetes fungi like rust, smuts are also present in this division for Basidiomycotina. Look at this. We have a white button mushroom, Biligundu anabe anta karitivi, Agaricus bisporus. It is available in the market. Another one is called pleurotus mushroom. Pleurotus mushroom. Okay. So this is also edible mushroom. In India, both the mushrooms are available and cultivated in large scale. So these fungi comes under Basidiomycotina, economically very important group. Another group of fungi called Lycoperdon, puffball anta karitivi. This fungus is also comes under Basidiomycotina. We will discuss the importance of these fungi later. Coming to the last division of mycota is called Deuteromycotina. Deutero means unknown. What is unknown in this group? Sexual reproduction is unknown. So those fungi which do not reproduce sexually, such fungi have been grouped under Deuteromycotina. That's why these fungi are called imperfect fungi. They are not perfect because perfect stage, sexual reproduction is not present in this group of fungi. That means these fungi reproduce only by mitosis. Hence, another name, mitosporic fungi, is also given to this group of fungi. We have penicillium, from which penicillin antibiotic is obtained. It also comes under this group. We have aspergillus. We have many biotechnologically important compounds are obtained from this fungus, aspergillus. All these fungi comes under Deuteromycotina. So these deuteromycetes are adapted to dry condition. They are adapted to land. They grow rapidly and they produce only asexual spores in large number. And many of them do cause food spoilage. Our food is spoiled by some of these organisms. And of course, some of the fungi are very important as food product, industrially very important fungi. Antibiotics are produced. So that's why these fungi are very, very important. So this is a just outline classification and some of the important group of fungi that we place in different divisions of fungi. Hope you have understood the basic aspects of the fungal world. So these are the essential divisions of the fungal world. Now let us move on to why we need to study fungi, why we need to understand fungal diversity, where they are distributed, what is the need for documentation. This is very, very important. So what is the fungal diversity? How do you define fungal diversity? Fungal diversity can be defined as the sum total of fungi and their genetic wealth or genetic pool together constitute the fungal diversity. That means total number of fungi which are present on the earth surface. Together we call it as fungal diversity. Every fungus is important. Every fungus and its genetic content is very important biotechnologically. That's why studying fungal diversity is very, very important because they play a very important role in biogeochemical cycle. They recycle all the nutrient back to the ecosystem. In that way, we need to understand fungal diversity. So these fungi, the total number of fungi can be estimated by cultural methods and that means and their enumeration, counting their numbers. So traditional methods are used. So whether it could be a soil or plant material, animal material, or any other natural material that can be brought to the laboratory, that can be serially diluted, okay? And these fungi, which are present in all these material, natural material can be isolated and you can count them exactly how many number of fungi are present. That is one method. With that, we have so far 80 to 1,20,000 fungi we have described and we have well documented. And there is a multi-billion dollars industry is also revolving around 1 to 2% of the fungi. Okay, so that's why they are very, very important. But very recently, we have molecular methods are available. By using molecular method, the fungal genomes can be isolated from soil or any other natural world. mRNA from fungi can be isolated. Ribosomal RNA can be isolated. Mitochondrial genome can be isolated from the soil or any other degraded environment. And thereby, you can also determine the total fungi which are likely to be present in ecosystem. So that gives you an idea about total number of fungi which are likely to be present in this world. So very recently, a very well-known mycologist, David L. Oxworth, in 2018, he estimated that there are 2.2 to 3 million fungi are likely to be present. So when he has estimated in 2018, he stated that 3.8 million fungi are likely to be present in our ecosystem. But he recently revised this data and he is now telling that 11.7 to 13.2 million species using DNA sequencing method, molecular methods, and other technique. He has estimated that 13.2 million fungi are likely to be present in this world. Where are those fungi? Because so far we have identified only recorded 1,20,000 remaining fungi where they are present. Okay, so that's why they are very, very important. And missing fungi. So these fungi may be present in the habitats that include soil, air, water, new geographic areas, extreme environments, so new vegetation types, so forest canopies, so marine environments, so atmospheric layers, so plant, insects. So if you analyze all these things, you are likely to get 10 to 20% of the fungi are new fungi. So based on this data, he revised this total estimate to 13.8 million fungi, which are likely to be present. And therefore, he suggested that these fungi should be identified and recorded, because they are the sources of newer antibiotics, new enzymes, plant growth hormones, immunosuppressant chemical, food colorants, bioinsecticide, and many other pharmaceutically important compounds. That's why we need to identify some of these missing fungi and we should document them. We should bioprospect them for our own benefit. So recently, we have in 1980s, biotechnology has helped up, the new discipline has emerged. We call it as a biotechnology. Along with that, we have a genetic engineering has also come up. We have a powerful molecular biology tool is available. And therefore, by using this molecular biology tools and the biotech revolution, the fungi are the important organism and we are interested, and there is a lot of scope for documentation and bioprospecting of these world fungi that is likely to yield very novel products which are likely to help us in many different ways. So that's why the new term bioprospecting is coming up. Bioprospecting is nothing but a systematic and organized search for useful products derived from fungi. So as I mentioned in another half an hour, I'm going to tell you why these fungal cells are very important, what are the important compounds that we have already recorded, and how multi-billion dollars industry revolving around these compounds. And in that context, the isolation, documentation, and commercialization and of these fungi for overall benefit to the society is very important. That field of study, we call it as bioprospecting. So this is very important, not only for botanists, it is for biochemists, biologists, molecular biologists, genetic engineers, who are all interested. All the biologists, even the people in medical field, they are also interested, because we have antibiotic, we have immunosuppressant, we have many other important pharmaceutical compounds have been isolated. They are very promising, which are likely to solve many of the problems which we are facing at the moment. So fungal bioprospecting is an important area in modern biotechnology, exploring the fungi and their genetic resources that can be used to create commercial product. So you can produce a products of high economic value using these fungal cell and fungal organisms. So applications are many. So with this one class, 1.5 hours time, I may not be able to give a complete justification, but I will try to give the essence of the role of fungi. They are very important biotechnologically, how they are important academically, not only academically, but they are also important biotechnologically. Coming to let us discuss fungi as model organisms. ಮಾದರಿ ಜೀವಿಗಳು ಅಂತ ಹೇಳಿ. What we cannot conduct certain experiment on highly evolved human and plant, but we can conduct certain experiment on eukaryotic cells like fungi. So one such best example is the Saccharomyces cerevisiae. Saccharomyces cerevisiae. So as you know that Neurospora crassa, and with the name Neurospora crassa, neurospora, because the spores which are produced by this fungus, Neurospora crassa, it looks like a nerve spore. Looks like a nerve cell. Hence, the name Neurospora crassa was given. It is an ascomycetes fungus, and you know that Edward Beadle and Tatum, they conducted a famous experiment on this particular organism, Neurospora crassa, and one gene enzyme hypothesis was proposed in as early as in 1958, for which they won the Nobel Prize in Physiology and Medicine. So that's why this organism, Neurospora crassa, is very important. It is used to understand elucidation of molecular events involved in circadian rhythm, epigenetics, gene silencing, cell polarity, cell fusion, cell development, as well as many aspects of cell biology and biochemistry. So that's why it is even now, it is called Cinderella of fungal world. It is very important organism. It is used for genetic studies, genetic recombination, tetrad analysis, many other aspects. So that's why it is an important model organism. Next to Neurospora crassa, we have Saccharomyces cerevisiae. It is another important model organism. It is a principal yeast used in biotechnology, physiology, cell cycle to understand the cell cycle, and it is used in food fermentation. Very variety of food that we are producing today, that involves Saccharomyces cerevisiae. Then we do produce several industrial chemicals, including alcohol. Where the Saccharomyces an important role, and we have this fungus is used to understand the fundamental cell biology research, and it is also used in neuroscience, bridging the genes to cognition. So that is another aspect of Saccharomyces cerevisiae. So another relative of Saccharomyces cerevisiae is the Saccharomyces pombe. It is called fission yeast. Saccharomyces cerevisiae is called budding yeast. Saccharomyces pombe is called fission yeast. Is an African breeding yeast originally isolated from millet bean. Now it is used to understand the cell cycle, mitosis, chromosome dynamics, epigenetics, DNA repair, meiosis, cytokinesis, mRNA processing. So that's why it is very, very important organism. As I mentioned, Aspergillus nidulans. It is a deuteromycetes fungus, but it is very important. In this fungus, a sexual cycle, an alternative to sexual cycle that operates, that is called parasexual cycle, a deviation from the normal sexual reproduction. Without meiosis, haploidy is achieved. So that is why a deviation, what we call amitosis occurs. As a result, these fungi undergo, recombination, and the variability due to recombination is very 0.001%. But these are the fungi which are asexually reproduced. In that asexually reproducing fungi, even 0.01% variability that is achieved through mitotic recombination is very, very important. So that's why this is a important fungus. Lot of research work is going on parasexual cycle and on this particular fungus. As I mentioned, Physarum falciparum is a slime mold. It is a slime mold. I told you it is used for isolation of mitochondria. Studying all the mitochondrial research work has been done on this organism, mitochondria isolated from Physarum falciparum. Dictyostelium discoideum, another slime mold used in morphogenetic studies. So it is a wonderful organism. And we have as I mentioned, the organisms looks like an amoeba. We call them as a social amoeba. If there is a threat, if there is an unfavorable environment condition, all will come together. All will come together and they take a state of a single-celled organism again, and from which a sporophore, fungal spore is developed. And these spores again germinate and individual amoeba, social amoeba are formed. So this is a very interesting organism. It senses the threat in the environment. and all the social amoeba come together. How do they sense that? It is a very interesting aspect. So that's why this organism is very, very important. And these are all the important organisms we are using as model organism to study various aspects of biology. Another group of organism, we call them as lichens. Lichens. Okay, kallu hoogalu anta kariತೀವಿ. They grow on stones and other rocky substrates. Okay, exposed rocks. Okay, today there are about 13,500 lichens are there. Kallu hoogalu anta kariತೀವಿ. 13,500, nearly 14,000 are there. They are distributed Arctic to Antarctic region. They are present. They grow on exposed rocks, soil surfaces in deserts, solidified lava, barks of trees, marbles of ancient temples, backs of Galapagos tortoise, backs, idiruttalla chippu adar mele beliyuttve, unwashed window panes, grave stones mele. Okay, there are so many very interesting organism. These are otherwise called biomonitors anta kariತೀವಿ. Biomonitor, because they disappear all of a sudden if there is a pollution in the environment. If there is a pollution, they disappear. Because the body of the fungus is made up of two partners. Eradu partner ide. One the fungal partner is called mycobiont. Another one is called algal partner is called phycobiont. So phycobiont prepare the food, give the food to the mycobiont. Mycobiont protect the algal partner and provide all other assistance for the algal partner. So together mycobiont and phycobiont, we call it as a lichen. So what happens when the sulfur dioxide concentration increases in the atmosphere? The sulfur dioxide kill the chloroplast which is present in the algal partner. Once the algal partner is killed, the mycobiont also gets killed. So that is why they disappear. So that's why they are called biomonitors. Very important group of fungi, and they are also very important to us because there are two types of lichens are there. Ascolichens. Some of the ascomycetes fungi associated with algae, they are called ascolichen. Some of the basidiomycetes lichens fungi, they associated with the algal partner, they are called basidiolichens. So as I told you, they are the first one to colonize the rocks. They produce the lichenic acid. These lichenic acid, they wither away the rock and they helps in pedogenesis. Soil formation takes place through the activity of lichens. Lichenometry, it is an important discipline. So by measuring the lichen, by identifying the lichens and by analyzing the carbon content in these lichens, by carbon dating technique, we can measure the age of the rock, age of the temple, age of the monument. So that discipline we call it as lichenometry. Lichenometry, and this lichenometry is used in geomorphological studies and archaeological studies, and there are variety of applications of these lichen. So we have very important, because lichens are source of food. They are the source of food for animals, and they are the very important component of ecosystem, and many drugs have been, very essential drugs have been isolated, and these drugs are very important, and people now mycologists are interested in isolating some of the pharmaceutical compound from these lichens, and they are likely to come up as drugs in the near future. Another interesting group of fungi, they are called mycorrhizae. Mycorrhizae. Myco means fungus, rhiza means root. They are called fungal roots. Look at this particular picture. This is a root system externally colonized by the fungus. Majority of our plants, whatever the plant that we see in our ecosystem, they are colonized by this group of fungi. They are called mycorrhizal fungi. They are mycorrhizal fungi, otherwise known as symbiotic fungi. So I have told you, many fungi live like symbiotic mode of life, mutualistic life, saprophytic life, and parasitic mode of life. So 97% of the fungi are saprophytic life. Another remaining fungi, they also lead a symbiotic mode of life, and there are others, they live a parasitic mode of life. So mycorrhiza is nothing but an intimate association between a root and a fungus. So roots of higher plant and a fungus, we call it as a mycorrhizae. 70% of the vascular plant that we see, they are all associated with mycorrhiza. There are different types of mycorrhiza there. Some mycorrhiza colonize the external surface of the root. They are called ectomycorrhiza. Some of them, they enter the internal cell. They are called endomycorrhiza. If they are present in orchid, they are called orchid mycorrhiza. If they are present in ericoid plants, they are called ericoid mycorrhizas. So there are very important group of mycorrhizas. They play a very important role in mineral nutrition. Their just association with the root system, they provide lot of minerals available to the plant. So they help the plant system to grow well. So mineral nutrition of the plant is achieved through mycorrhiza. Water balance. What plants cannot absorb water, mycorrhiza helps to absorb water from soil particles. They also protect plants from toxins which are secreted by other chemical or biological agents. They do protect the plants against pathogens, disease-causing organisms. So very important role played by mycorrhiza is conversion of inorganic phosphate to organic phosphate. Inorganic phosphate. Inorganic phosphate is an elemental phosphate. Plants cannot utilize this elemental phosphate. So plants will only use ionic phosphate, PO4, not P. So therefore, the phosphate solubilization is done by the mycorrhiza. So that is why their very important role in soil is phosphate solubilization. So that plants utilize this phosphate. And you know the importance of phosphate. Phosphate is the important backbone of your DNA and many other macromolecules in our body. So therefore, these mycorrhizas are considered as key components of our ecosystem. So they evolved along with the plant and they live with the plant, and we cannot separate them plants, and we cannot grow them in the laboratory. So that's why they are very important partner of our plant system, and very well-known forests of our world are all associated with mycorrhiza. If the mycorrhiza disappear, most probably the well-known forest will also disappear. So that is the important role played by mycorrhiza. Another important group of fungi, they feed on soil, nematodes. Nematodes are small organisms. They are called eelworms. They are present in they belong to the animal world kingdom nematoda, and they eat away the plant roots and cause many diseases. So how to control these nematodes? Very, very difficult. We have to, inject nematode killing chemical pesticides into the soil. If you inject these chemical, all other microscopic organism, all the other living organism will also get killed. So what is the solution? Okay. So we have mycologists have identified nematode eating fungi. Look at this particular picture. Many nematodes are entangled, squeezed by the fungal mycelia, filamentous thread-like mycelia. Okay. These fungi are called nematode eating fungi, nematophagous fungi. These include Catenaria anguillulae, Nematoptera gynophila, Arthrobotanus oligospora, Dactylaria, Verticillium, Chlamydosporium. These are all the important fungi. They feed on these soil-borne nematodes and control the nematode, and thereby they protect our cropland from diseases. So that's why these fungi can be multiplied, and we can release them into the soil so that these fungi will take care of nematode problem in our agriculture fields. So look at this particular picture. It will tell you this is the nematode. Now look at the fungus. How this fungus has entangled the nematode, and it is with the help of ring-like structures. These are all the ring-like very sensitive rings. Okay, they squeeze the nematode and obtain the nutrient from the nematode and kill them finally. Some of the other nematode, they enter the mouth part of the nematode, colonize the entire body of the nematode, and finally they kill the nematode. So that's why they are very, very interesting group of fungi. They help us, particularly agriculture, in growing very important croplands. Another group of very interesting fungi in the fungal world are insect-eating fungi. You know that many insects do trouble us. particularly farmers. They eat away the leaves and other edible part of the plants, and controlling some of these insects very difficult. We are spraying in an annually 30 to 35 different chemicals to control these insect problem. In cotton only, we spray 30 different types of insecticides. But very recently, we have identified some of the fungi like Metarhizium, Bavaria bassiana, Hirsutella thomsoni, Verticillium lacani. These are all the fungi. They feed on insect. And these fungi are known to produce an enzyme called chitinase enzyme. These chitinase enzyme dissolve the exoskeleton content of insects. And once the exoskeleton content of the insect gets dissolved, the water starts evaporates from the body and insects will die because of dehydration. So that's why they are very important biocontrol agents of insect pathogen, insects. Okay. So nowadays, many farmers are using these insect eating fungi. So their cultures are available. Only thing is we need to release them in the environment so that they will take care of the pest problem very effectively. In addition to this, many fungi also cause diseases in plants. So some fungi parasitize another fungus. These are all called mycoparasites. So some of the fungi like Trichoderma viridae, Trichoderma arzianum, this is a very important group of fungi. They control another fungi which are disease causing agent. For example, Fusarium oxysporum, it is a wilt disease, Soragu roga. ಟೊಮ್ಯಾಟೊ ಈ ತರ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಸೊರಗು ರೋಗ ಕಾಸ್ ಮಾಡುತ್ತೆ. ಫ್ಯುಸೇರಿಯಂ ಆಕ್ಸಿಸ್ಪೋರಮ್ ಇದು ಒಂದು ರೀತಿ ಶಿಲೀಂಧ್ರ, ಇದು ಸಾಯಿಲ್ ನಲ್ಲಿ ಇರುತ್ತೆ. ಬಟ್ ಇದನ್ನ ಕಂಟ್ರೋಲ್ ಮಾಡೋದು ಬಹಳ ಕಷ್ಟ. ಫಾರ್ ದಟ್ ವಾಟ್ ಯು ಕ್ಯಾನ್ ಡೂ ಇಸ್ ಯು ರಿಲೀಸ್ ದಿಸ್ ಫಂಗಸ್ ಟ್ರೈಕೋಡರ್ಮಾ ಆರ್ಜಿಯಾನಮ್ ಇಂಟು ದ ಸಾಯಿಲ್. ಸೊ ದಿಸ್ ಫಂಗಸ್ ವಿಲ್ colonize. It will over parasitize Fusarium oxysporum, Rhizoctonia solani, Fusarium culmorum. These are all disease causing agent. So these fungi will control these fungi naturally without spraying any chemical pesticide or chemical fungicides into the soil. So that is why they are very important biocontrol agents in agriculture. So now they are commercially Trichoderma cultures are available. We can mass multiply them. And we can release them into the nature. Farmers are using this fungi in their field to control wilt disease in tomato and other agriculturally important crops. As I mentioned, the fungi play a very important role in decomposers, decomposition. So we have saprophytic fungi, cellulose degrading fungi, lignin degrading fungi, and they are very important component of compost making. Every day, every house is releasing 2 to 5 kgs of waste. If this waste is not treated, it will create a huge pollution problem. So that's why these fungi play a very important role as fungal decomposer, and that's why these fungi are very, very important to us. 97% of the fungi, they play as a saprophytic mode of life. They degrade the organic that into an organic matter. Many fungi do cause diseases in humans. Some of the important ones are the fumigate, Aspergillus fumigatus, okay, Histoplasma capsulatum, blastomycosis, dermatitis, skin problem, Coccidioides immitis, Candida albicans, mouse thrush, mouse thrush, Candida albicans, Trichophyton, skin problem, Microsporum, skin problem, Epidermatophyton, Cryptococcus, yeast. These are all the important fungal pathogens. They do cause diseases in humans and very life-threatening sometimes, but their percentage is very, very less. Not many people are susceptible, all are resistant. Only those individuals who are immunocompromised, they are likely to be infected by this, for which we have a medicines are also available from the fungal world, and let us try to understand. So we have if the Histoplasma capsulatum causes a disease called histoplasmosis, similarly blastomyces, dermatitis class, blastoplasmosis, cryptococcosis, cutaneous mycosis, skin infection, okay, pneumocystis, pneumonia. This is I will tell you a little bit more about this later, particularly in HIV infected people. This very important pneumonia disease. Pneumonia disease caused by Pneumocystis carini. It is very important. Candida albicans cause candidiasis, thrush, Aspergillus fumigatus, aspergillosis. It's a lung infection. Okay, so these are the important human diseases for which we have medicines are also available. So this is just to show that how human body, though human body is harbor very important fungal species, some of the fungi do cause diseases in human body and sometimes life-threatening also. Very recently, during COVID-19 period, SARS-CoV-2 responsible for COVID-19, during that particular period, mucormycosis, the black fungus also responsible for death of few individuals, but we quickly detected the causative agent, and we have medicine was also available, and we could able to control the disease. This fungus, mucormycosis, was caused by the fungus, zygomycetes fungus called Rhizopus varice. Okay, and very few people were affected, and quickly we have detected and we have treated them, and we could able to save many people. And this is the disease was known as the mucormycosis or black fungal disease. As I mentioned, Pneumocystis carini. And this is one of the Ascomycetes fungus. Without knowing this fungus, people consider that pneumonia was caused by a protozoan. So therefore, they started treating only protozoa. Protozoan treatment was given. But later, we isolated this fungus from the human lung system, and finally, we identified it as Pneumocystis carini. How we could able to do that? Because plant animal cell contains their cell wall, cell membrane contain cholesterol, but the Pneumocystis carini, its cell wall contain ergosterol. All fungal cell membrane, cell wall contains an important alcohol called ergosterol. Based on this, we disproved the theory that pneumonia was caused by protozoa, particularly in HIV-infected individuals, and now it is considered as an Ascomycetes fungus, Pneumocystis carini. It was responsible for death of millions of AIDS patients in North America. Even now, this is a serious problem in other part of the world, and we have to be very careful about this. Fungi not only cause diseases in humans, but many fungi also cause diseases of crop plants. So agricultural crops are also affected by fungi. 70% of our major plant diseases are caused by fungi, and there is, as a result, 50% of the food that we grow is lost to fungal infection. Either it is during before harvest or after harvest. Some of these fungi are known to cause a variety of diseases. Some of these pathogens are called necrotropic pathogen. That means first they enter the plant cell, they kill the cell, and then they obtain the nutrient from the dead cell. On the other hand, there are very clever fungi. They never kill the host cell. They live within the living cell and draw continuously the nutrient from the host crop plants. So there are many diseases are caused by fungi, which include fruit diseases, vegetable diseases, diseases of plantation crops, diseases of stock rot diseases, market diseases. Even after harvesting the crop, when we store the food, even during storage, many fungi attack some of the stored fruit products and bring about spoilage. And as a result, certain toxic substances are accumulated and render the food unsuitable for human consumption. So that is why 50% of the loss is due to fungi which parasitize agriculture crops either during the pre-harvest period or after harvest period. So these are all the very interesting aspects of fungi, how fungi negatively play a very important role as human parasites and also plant parasites. Now, many fungi have been identified and they have been exploited in biotechnology. After the emergence of biotechnology, now we are using fungal cell, yeast cell as factory cells to produce products of high economic value. That field of study, we call it as biotechnology. Fungi have been exploited very well, and biotechnology is an integrated use of biology and engineering principle to isolate products of high economic value, so that these compounds will benefit human and society immensely. So look at this. These are all the areas how fungi have benefited us. One is the mycoprotein production, fermented foods, fungal metabolite like organic acid production, antibiotic production, immunosuppressive agent production, anti-cancer drugs. I will give you one or two examples for this also. Enzyme production, vitamin production, hormone production, then vaccine production. Today we are using Saccharomyces cerevisiae yeast to produce vaccines. Your hepatitis B vaccine is produced from Saccharomyces cerevisiae. Then edible oil. It is nothing but a glycerol. So no need to get oil from only from plant source. We can use the yeast to produce oil. That is called edible oil. And we have alcoholic beverages like wine, beer. It is a multi-billion-dollar industry. Several agrochemical, biopesticides are produced from fungi. We have very recently fungal pigments are likely to replace our textile industry. We have alternative food sources. Variety of mushrooms we are cultivating. Mass production technology has been developed, and many of fungi we are using as pest control, disease control agent, as biopesticide. Some of the interesting aspects we will discuss one by one. One of the interesting aspect of fungal world is mycoprotein production. Mycoprotein is nothing but fungal protein. You know that third world is suffering from protein malnutrition. And government of India after independence, particularly in 1970s and 80s, we started growing pulses to provide proteinaceous diet for Indian populace. But we were not successful. Okay, even now, protein deficiency diet we are all eating. So one of the alternative to overcome protein malnutrition is looking for some other alternative. Here, the fungal world, we can produce a very interesting protein. It is called mycoprotein, otherwise called meatless protein. This protein is isolated and obtained from fungus called Fusarium venenatum. It is a Fusarium venenatum. It is a fungus. It can be grown in a fermenter. The mycelium filamentous thread-like structures can be harvested, and RNA content in the filamentous thread can be removed, because RNA is not good for us. Okay, uric acid content will be increased in our body. So therefore, when you remove RNA content, this Fusarium venenatum protein, filamentous thread will serve as a protein-rich source. It is a future protein. It is a solution to the future protein shortage. And McDowell company, McDowell company in England has produced this compound product. Now this commercially available product. This is one of the best application of fungal world, fungi for human benefit, mycoprotein production. So you know that the fungal, particularly fungi like Saccharomyces cerevisiae, we are now using to produce ethanol called gasohol. Okay, gasohol is the fuel. Now we are all interested. Gasohol is nothing but 96% ethanol and 4% glycerol. We have in Brazil the sugar cane. Sugar cane is grown to produce this gasohol, a fuel. They are not using your petrol and diesel, all these, fuels to run engines and other automobile industry. Now they grow sugarcane. They have a national alcohol program. In that, they produce a molasses. Through molasses, they produce ethanol, and they mix with 4% glycerol. Now it is sold as gasohol, and it is used to run all kinds of engines. Using gasohol in engines, it is very interesting because it burns completely. Burning is pollution free. So pollutants will not be eliminated. That is one of eco-friendly. So that is one of the interesting aspects of Saccharomyces cerevisiae. Okay, so where we can produce industrially important alcohol, which is likely to be the future fuel for the world. So similarly, Saccharomyces cerevisiae is also used for beverage and food production. Whole world is talking about two important things. One is wine, the other one is beer, the third one is the bread. Bread, and these are the two important aspects. It is a multi-billion dollars industry, okay, revolving around single fungus, Saccharomyces cerevisiae. It is an ascomycetes fungus, okay. So therefore, very interesting. So look at that, how a single fungus created so much of employment, so much of industry, so much of economy, so much of food for us. So in that way, you need to look at the fungal world, and you need to look at the role played by the Saccharomyces cerevisiae, how it is helping us to future, shaping our future. Wine fermentation, we have very interesting different strains of Saccharomyces cerevisiae are available for industrial alcohol production. We can use different types of industrial waste, agriculture waste that is available. You can convert that waste into alcohol. So in addition to Saccharomyces cerevisiae, variety ellipsoides, this is called wine yeast. In addition to that, Saccharomyces Saccharomyces carlsbergensis, these are all other species. We can use them for industrial alcohol production. So today we have more than 200 fungal pigments are also being produced through cultivation of fungi. Look at this. These pigments are nothing but carotenes production from fungal cell, and they belongs to zygomycetes, and they also belongs to mucorales, which includes produce different types of colored substances. These can be used, okay, these are all the important pigments, and these can be used in food and beverage industry, healthcare industry, cosmetic industry, textile industry, and many are waiting in the pipeline. Many food enzymes are also produced using fungal species. In addition to that, look at this. This slide will tell you these are all the important fungi that we have used to produce enzyme, and these are the applications of fungi in different industries. In addition to that, several organic acid, your citric acid, gluconic acid, itaconic acid, chemical synthesis of these acids is costly, but by using some of these fungi, we can produce a high concentration of organic acid, which are being used in our laboratory. Food additive, vitamin B12, riboflavin, okay, even biopolymers, they are a biodegradable plastic like xanthan gum, pullulan gum, they can also be produced. So therefore, these are considered as they are likely to replace the synthetic plastics in the years to come. In addition to that, mushroom, the variety of mushrooms have been isolated, and these mushroom kits are available. And we have in India button mushroom, and then padistra mushroom, oyster mushroom, chippu anabe anta karitivi. These are all very popular. So it is a multi-billion dollars industry. We are all very recently in India also we started eating very less carbohydrate content, sugar less, zero sugar, particularly for diabetic patient. It is a cholesterol-free food. That's why these are all very, very important. People are eating and maintaining their health very well. In addition to that, there are anti-tumor properties. If you eat mushroom, the aging can be postponed. Aging can be postponed because they contain new peptides in it. That's why they are very, very important. These are all the other important applications of these fungi, okay, commercially. There is one mushroom called Ganoderma. And medicine prepared from this fungus, Ganoderma, treat 12 different diseases of human being. It is available in the market. Okay, so Ganoderma capsule is available. It is a magic herbal medicine. It will treat 12 different types of diseases. That is the interesting part of fungal world. So Cordyceps sinensis is a tonic which is available. Okay, it is produced from the fungus Cordyceps sinensis. Okay, and these are all the marketability of this particular product, and it is produced commercially, and these products are available. In addition to that, there are fungi which live inside the plant cell. They produce several chemicals, and these chemicals have been exploited very recently as anti-cancer drug. Okay, you know the importance of antibiotic, penicillin antibiotic produced from this fungus, Penicillium chrysogenum. We have griseofulvin antibiotic. Okay, it is from the fungus again. Okay, so your skin disease, fungal diseases can be cured by this griseofulvin antibiotic. A fungus produces antibiotic which cure another fungal disease. They are very, very interesting. This is very interesting. So I told you endophyte, some fungi plant present inside the plant. One such fungus is called Taxomyces andrinae, which produces a chemical called Taxol. Is an important anti-cancer drug. It is used for the treatment of breast and ovarian cancer, in the current market. So Taxol is it is produced from the fungus nowadays. That is why several anti-cancer drugs are also isolated from fungi. So earlier it was isolated from Taxus baccata plant. Now the fungus Taxomyces andrinae produces this Taxol drug. It is used for the treatment of cancer. Steroids, many steroids. One important steroid is called cortisol. It is secreted by the adrenal gland. If there is insufficient secretion of this hormone, rheumatoid arthritis will result. Now the fungus Rhizopus arises produce this, transform this precursor into a cortisone hormone, and the cost is very, very cheaper now. So vaccine can also be produced using Saccharomyces cerevisiae, your hepatitis B vaccine, hepatitis HPV vaccine, SARS-CoV-2 vaccine. All these vaccines are now produced in Saccharomyces cerevisiae. This is how these fungi are very, very useful to us. I will, because of time, I may not be able to tell in detail. Fungi can also be used to treat polluted environment, wastewater treatment, heavy metal treatment, pesticide can be treated, textile industry waste can be treated, leather tanning industry, we can use some of these fungi, we can treat the waste, that particular field is called mycoremediation. So these are all the pigment, organic acid, enzyme, secondary metabolite, fungal metabolite produced by some of these fungi. Look at the applications. So this PPT is available. You can ask for it. You can read it later. You will understand that. So this whole fungal biodiversity is under threat because urbanization, habitat destruction, deforestation, mining, environmental pollution, many fungi are dying. That's why time has come. We need to conserve these fungi. Safeguarding fungi from future exploitation is what we call conservation. We need to protect them. There are two different methods, in situ conservation and ex situ conservation. In situ means original habitat must be conserved. Bioreserve parks, something like that. Ex situ means certain soil, certain area, we can go there, isolate the fungus, preserve them in the laboratory. That is called ex situ conservation. Inside the lab, we can preserve them. By preserving that, we can preserve the ecosystem, we can maintain genetic diversity, we can prevent any fungal species going to extinct. So therefore, new exploration, isolation, identification, documentation, germplasm collection, molecular conservation, education and awareness. I'm teaching all these things. You must teach your student that fungi are very important. They are important component of our ecosystem, important component for us, for our future. In that context, you need to bring awareness to the student so that fungal diversity, fungal world can be protected. So that is why with these few remarks, I request all of you to go with this message that the third kingdom fungi, which include variety of different organisms, they are very important, particularly for biologists, molecular biologists, biotechnologists, for everyone who are involved in